Hello everyone, I am Dr. Somnath Ghatar. I am application specialist from Carl Zeiss, India, and today I will be demonstrating on a confocal laser scanning microscope from Carl Zeiss. So as you can see here, the model of the confocal laser microscope is LSM 980 I will be talking of. In this module, we have a microscope platform on which the confocality with the laser light and the light sources with a light cell attachment has been given. So to start with, this is an inverted microscope frame, and in this inverted microscope frame, as you can see, we have basically like a light source given. So three light sources are given. One is a halogen light source, one is a metal halide light source. The laser light source is given here. So if you start from halogen 100-watt light source has been given. This light source is mainly used for transmitted light studies for morphological imaging, and this light source has a shutter, inbuilt shutter, to control the inflow and out of the light. So the light from here comes through the condenser and falls onto the stage. On the stage, the sample in the form of a glass slide has been kept. In this stage is a motorized stage, so the XY movement is fully controlled by the instrument itself. Just below the stage, we have the nose piece on which the objectives have been mounted. So there are 6 positions available, and out of 6, 5 objectives has been given. The objectives are 10x, 20x, 40x, 63x and 100x. 5 objectives given. 10, 20, 40x are dry objectives, and 63x and 100x, these two are oil immersion objectives. For future upgradation, there is one more position still available. Just below the objectives, we have the fluorescent turret. So basically the fluorescent mirrors are there, which restricts the emission light and the excitation wavelength with a combination of a dichroic mirror, and this light comes here for the viewing purpose in the eyepiece. So after talking of the microscope, we will come to the desktop where the Zen 3.1 blue edition has been given. We will open the blue edition here, and we will get a guest user interface like this. So in the guest user interface, under locate, we have the entire architecture of the microscope in the form of a light path. So as we discussed, we had the halogen lamp here. So halogen lamp, you can control it through the software, and the intensity of the light can be increased or decreased based on the control unit from here. So there is a shutter which controls the light. So if you can see, the light now falls onto the sample through the objective, and we can see it here. So this shutter actually controls the light falling onto the sample or not. After this is the condenser, which is again a motorized condenser. This is the stage, and on the stage the sample is there. Below the stage, the objectives have been positioned. So 6 position is there. So 10x 0.45 numerical aperture, 20x 0.8, 40x 0.95, 10, 20, 40 are dry objectives. 63x and 100x is 1.4 numerical aperture, and they are oil immersion objectives. Below the objective, then we have the filter turret. So in the filter turret in the system, for bright field or transmitted light, this will automatically pass through the analyzer and the DIC prism that is placed. And if we click here, it will be for the morphological studies. So, and through this, it will pass through the tube lens below, and then it reaches to the eyepiece, which is here. So eyepiece is 10x, and the objective multiplied the 10x of this will give the total magnification of the sample you are looking for. Now, if we are looking for a fluorescence sample, for fluorescence sample, this is the light source. So HXP 120 volt, which is this particular box, 120 volt. It is totally controlled through the software. As we can see here, the intensity can be totally controlled through the software. See here, light is coming onto the fluorescence filter turret, but it is not going ahead of it. So from the filter turret, based on what I want to see, depending on different bandwidth or the excitation and emission composition, the filters has been given. For example, if I want to excite it in the UV range and want to have an emission in the blue range, I will use a DAPI filter. DAPI is usually a dye which is used to stain for nucleic acids. It's commonly used as a counter staining technique. So if I want to see DAPI, I have this option with a band pass of 335 to 388, that is the excitation band pass, a dichroic mirror of 395, and emission band pass of 420 to 470 nanometer. But if I'm looking for something like green, Alexa 488, then or a EGFP, green fluorescence protein, in that case you have to use green fluorescence this filter composition, which has a band pass, excitation band pass of 450 to 490, as you can see here, with a dichroic mirror of 495, and the emission is collected from 500 to 550 nanometer. Similarly, for the red, if we want to select red, in this red also 533 to 558 is the excitation band pass which is allowed. There is a dichroic mirror, so anything above 570 will be coming to the collection window, and which is from 570 to 640 nanometer. That bandwidth will be collected for the red. So right now, if I see on the green, I can see the green light the moment I click here. So for the green filter, the light which is passing is the blue light, but after passing through the filter, the emission from the sample, the green light, as you can see, is being visible here. So here you can see the blue light is falling onto the sample, but here the visible color will be green. Once we have done this, we need to do a quick focus adjustment. So by setting the eyepiece here, I am going to focus based on these two knobs. So this knob is actually for adjusting the Z position, see whether the sample is in plane of focus or not. Once there is a joystick here, with the joystick I can change the XY movement controlled through the joystick out here. So now once I have positioned, focused my sample, I can see it, the signals are good enough, I would like to have acquire those images in a laser mode. So right now we have been working with either the halogen lamp or a mercury halide lamp. Now when we will go to the acquisition part, we will be using the laser light sources. Now we'll move to the acquisition part, and in the acquisition what we will do, we will go to the smart setup. So in smart setup, as you can see, I have selected 3 dyes, blue, green and red. So now blue, green and red, that will be like Alexa 488, that is for the green, Alexa 594 or 561 according to your dye. So I have selected 594, and a DAPI which will be excited by 405 laser. So once I select, I have a window here, I can select as many dyes as possible. So I can click here, and from the database there is list of dyes available in the drop down menu. So as you can see here, in the drop down menu there are mostly commercially available dyes have been exhaustively listed here, and based on my dye I can select any dye and add here. So after I add, the system automatically will give you 3 options. You want speed or you want signal. So if you are interested in signal, you click onto the signal, and if you want to have the fast imaging, you have a fast imaging, you have the best signal, and you have the smartest one. So by convention, getting the best signal should always be the objective when you are working with a fluorescence sample. So now if you say OK, the system will automatically configure based on the 3 dyes which you have selected. So all the laser selections, the dichroic mirrors, the main beam splitter, they will all align based on my selection of the dyes, and it is fully motorized. So you just have to click. With a click of few buttons, it is ready. So first thing is, we have to have a localization image, just a viewing image, whether my sample after focusing, how does it look with the given condition. So I will try to optimize taking one track at one time, that is one channel at one time. So first I'll see for the red, And with the red, if I just click on the 594, so my 594 lasers, you can see here the list of lasers which are available. So I have to first see whether all the lasers are on. In this system, there are 7 lasers available. So starting from 405, 445, 488, 514, 561, 594 and 633. So all these lasers are available, which covers the entire visible spectrum, starting from near to UV range to near to IR range. So that entire visible spectrum is covered by the 7 lasers, and based on this availability, we can have the freedom of selecting any dye which is present either even if it is in the NIR region or even if it is in the visible spectrum. We have that freedom here. So first I'll optimize this 594 or 561 rather will be the laser I am looking for. So this 561 laser I am using here, and this 561 nanometer 0.2% is the laser intensity I am using. Now as this is a confocal system, so the instrument has to be optimized based on the pinhole aperture which is internally present at the conjugal focal plane of this microscope. Now at the conjugal focal plane, this the pinhole aperture size or the diameter entirely depends on the point spread function, which we say here as 1 ARI unit. So this ARI unit will always give an impression that this is a confocal image I am acquiring it. So I have to click on this 1 ARI unit so that it is in the confocal mode, and then there are few parameters which will be like for the optimization of the detector. So these are all optimized. I will quickly see how my image looks like with a continuous mode. So once in a continuous mode, I have to click it, and then this is what I am getting. So either this signal is not being excited by just 0.2% laser. So first thing is, I will just try to change the focus and see whether my sample is in focus or not. So once I quickly move, I see I get some signal here. Okay. So once I start getting some signal, what I'll do is I'll try to increase the laser power slightly up so that my signal intensity improves. So you see, with just 0.2 to 0.9% of the laser increment, you see the signal intensity has improved quite a bit. I will try to see the focus position again. And once I am convinced that this is the best focal plane I have achieved, what I do is I can also check with the range indicator. So this range indicator will give me an optimization whether my detectors are getting saturated or not. Just a click here will give me an impression. So this grayscale is basically the signal, the black one is the background, and if there are red dots, then the signal saturation signaling will come. But the red dots is not available, you cannot see here. So we can still take the liberty of increasing the detector voltage slightly so as to improve the signal to noise ratio to a certain extent, or better than the previous one. Right. So once we have optimized for red channel, we will quickly go and see the green channel. So in the green channel, what we will do is similar 488. So 488 laser will be using to excite this 0.2 and 650 volt. So we'll simply start with that, and let's see how we are getting this image. So this image is also good. I'll just check the Z. Okay, and histogram adjustment, and I can see the image quality here. It's perfectly in focus. The range indicator says there are no saturation points, so I can immediately stop this and optimize the third channel, which is my DAPI. So DAPI will be excited by 405 laser power. So with 405 laser power, I'll just click here, and I can see the signal which can be like slightly better with increasing the laser power, and this is so now all the three channels has been optimized, the DAPI, the green, the red, and once this is optimized, what I'll do is I'll select all the track. So once all the tracks are selected, I want to take a quick snap of all the channels. So I'll still take a snap, and once I take a snap, I will get all the channels, single 2D images of all the channels. So as you can see here, this is for the red, this is for the green, and lastly for the blue. So I have got 3 channels because in this sample I have 3 dyes. So based on what dyes you have in your sample, you can simply select the list of dyes, select prepare tracks, and then if you optimize each and individually, you will see all the tracks like red, green, blue, and this is a merged one. So a merged one will have all the 3 signals in the same place. You can see the merged one, which is having red, blue, green, all the 3 channels in the same place. Okay, once we have done a multi-channel, we would definitely like to optimize and use this system for the Z resolution, because confocal always is known to give a very good Z resolution. That is why we will be taking a Z stack and we will try to do optical sectioning of a sample which is very thick. So for example, if it is a tissue or it is a part of a stem, a TS section of a stem, that can be virtually sliced by using this system. You don't have to go for a microtome sectioning and all. So how we do it? We have optimized these 3 channels. So once we have optimized, we will just take one channel, one track, and based on that we will just select a Z stack. So in the Z stack, what this window opens up. In this window, we will just go in the continuous mode and try to see what is the focus of the sample. So this sample is in perfect focus. What I have to do, I have to go out of focus in both the directions, plus Z as well as in the minus Z. So for doing that, what I'll do, I'll change the focus from here, and as you can see here, the signal is getting lost and it is completely lost now. See, the signal is getting lost. So I will mark this position. So the Z value is marked at this position, which is the start position here, the first one, and then I will go in the reverse direction so that I get to see my sample. So now I am back in the focus. Once I am back, I will move to the other side. Move to the other side, stop the laser and set the parameter. So you can see here from this 1528 to 1575 is the range which has a thickness of 46.18 micron. So the sample is 46.18 micron thick, where the software will automatically tell based on the optics I am using, the objective I am using, the numerical aperture, the resolution capacity of that objective. So based on that, what the software can say, you can have a resolution in the Z-axis of 540 nanometer. So that is 0.54 micron, so which you can see here. So the moment I click on the optimized part, you see the 86 slices, immediately it will keep on. So once I have decided the plus Z and the minus Z of the sample, what I'll do, I'll select all the 3 tracks and then I will start the experiment of taking the Z. What it will do, it will take all the Z positions of each of the channels. And as you can see here, so what I'll do is, it will first go in the red channel, take Z position from plus Z to minus Z, complete the entire Z stack, go to the next channel where it will take the green. In the green it will again cover from plus Z to minus Z, which is already predefined, and a similar process will also be done for the DAPI, that is the blue. The Z is being taken, and as it is acquiring more and more, you can see the progress happening. So as you can see, different Z positions in red channel is being acquired, and for acquiring hardly 3 minutes is being used, so that time it is taking. So there is always a histogram below. So this histogram will tell you the signal intensity. As you can see, if I right click here, it is being taken at 16-bit. So this 16-bit distribution you can change also, but depending on the signal to noise ratio, this will automatically come. So it is like most of the time it is either 16-bit or you can keep it at auto. So once the red is being taken, now the green. So as you can see the green channel, you can select the green. So the experiment process, you can see here what is the time it is being taken. The entire experiment process can be visualized from here, and at the same time what is the percentage of the progress of the experiment can also be seen, or keep a real time tracking. You can do it from here. So you see 96%, this is about to get completed. The DAPI So I have DAPI, FITC all together. So you will see here, so red, green as well as blue, all the 3 channels I have got it. And in this, if I go to the gallery, these are the slices. So you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, and 1, 2, 3, 4, 5. So this is how much? If you see in the Z, it is 47. So 45 and 2, 47. So 47 slices we are getting for each channel for red, green and blue. And if we do orthogonal view, so in orthogonal view we can see each and every, like this is a XY plane which we are getting it, but this is a XZ plane which will give the thickness idea, and this is a YZ plane. So in YZ plane, if you want to measure the thickness, what you have to do, you can simply measure it using a 3D. You can say I want to measure the distance between 2 cell outlines. So there are 2 cells which are lying close to it. You want to see if there is a gap or not. If you want to measure that distance, what you have to do is you have to simply click start. So as you can see here, we can also have the set end position, and in the set end position if you click on the other side, so we can say this is my set end position, and based on the start and end we can have the distance which is 6.348 micron. So in the Z, what is the thickness, that also we can see here. I think it is not that clear. So if you can see here, this is the measurement we did. You can see this yellow line. So this is basically the 2 cells boundary touching to each other. If you want to measure this thickness, so with that thickness is around 6.348 micron. So this is in the Z plane also we can measure, not only in the XY plane. And if you want to have a 2.5D, so which will give impression whether it is how much raised from the flatness. So as you can see for the red and green, you can see the distribution here. For red as well as for the green, for the blue, sorry, you can see the distribution here, and then you can move around in different Z position to see for different channels. You can also have a 3D option. So a 3D is powered by Arrivis. Arrivis Arrivus is a software which is usually like used for high-end 3D imaging. So recently Arrivus has been powering the 3D component here, and if you can see this image, so you can have a 3D aspect of it. So this is a thick sample, and if you want to do 3D, you can rotate it in different angle, and then you can have the image. Apart from this, if you want to further process this image, so I want to further process this image, I can go to the processing part, and here I'll say simply I want to deconvolve this entire experiment. So based on this, I'll select the deconvolution, and once my deconvolution is selected, you'll see the progress because it has multiple Z positions. So as you can see, after deconvolution, this is my deconvolved image, and in this deconvolved image I can split and see for different. So based on this, I can also play around with the histogram to make it little look better. So you can see all the three, and it can be for the three different channels. Now coming back to acquisition, so this is a multi-channel image which we have taken. We have taken few more multi-channel images like this one. So you have taken all multi-channel images. There are also multi-channel images like this. So red, green, blue, all the three have been merged together at the same time. So this is when you have multiple dyes and you want to get an image. But before that, you have to also understand that the order in which, so higher the wavelength, lower is the energy associated. You have to optimize starting from the higher wavelength or the highest wavelength, and based on that you have to go down till the lower wavelength which has the highest energy, so that your sample doesn't bleach. Second thing is, you also have to select the detectors which are available. So in this system there are detectors available. Basically two types of detectors are there. First one is a multi-alkali PMD detector, and the other one is a higher sensitive one that is called a gallium arsenide phosphate PMT. So that is basically a gallium gas detector we say, or a high sensitive detector which has a quantum efficiency of 45% compared to the multi-alkali detector which has a quantum efficiency of 25%. So as the quantum efficiency increases in gas detector, the sensitivity increases. So for weaker signal, it is always advisable to use a gas detector compared to a multi-alkali detector. The lasers, as I previously mentioned, so all the lasers, you have 7 lasers, all are diode lasers. So it is like on and off. They have a life of 10,000 hours, so there is no worry about the laser power. So once you have done multi-channel, we have also done optical sectioning using Z. Now we will do for different like time series. But if we do time series, so you have to click on the time series, and this time series options will come. Time series is important when we are doing with live samples. So with live samples, if you can show. So for live cell, as you can see, this is an attachment which is basically a stage top incubator. So this is a stage top incubator, and this is the cover lid. So on the cover lid it's a glass, and you can have a temperature control for the cover lid also. And below the cover lid you have this platform. This platform comes from Picon. So this has an internal chamber which you can see here, a Petri plate, 35 mm Petri plate is placed onto it. You can also put lab tech chambers. So this cover goes, and as you can see here, it sits onto this notch, and this entire attachment can sit. So you can see this sits on top of it, and then you have to place the lid which is covering, and this will go in the groove. So this closed, the entire sample along with this entire chamber is closed. Now we can have the condenser, the pillar can be down with the condenser up so that the condenser doesn't touches the top of the glass. This particular tubes, you can see here there are a couple of tubes. So this tube comes from here, So we have the CO2 coming from the cylinder, which is passing through a sterile distilled water or preferably milk, and the humidized CO2 which is coming from those cylinders. So you can see those cylinders, the CO2 from the regulator is coming, it is coming onto this box here, and from here it is going to this water chamber. From the water chamber the CO2 is getting humidized, and through this tube, it is coming onto the sample, to the incubation. Now this incubator, stage top incubator, can maintain the temperature of this lid. It can also maintain the temperature for the bottom, and the circulation is uniform. So the similar thing we can do it from here. So once the live cell setup is ready, before anybody is coming for live cell setup, this has to be on, and the stage top incubator has to be there. So, and this in incubation, you can see the window. So here you have to set the temperature. So usually it is kept at 37 degrees. So you have to set the temperature for the inserts. Okay, so as you can see in the incubation here, there are set of parameters. Basically 37 degrees is the set temperature which is there. You can also have different set points according to your experiment requirement. And this is the measured temperature. So this is the current temperature which you are having in the system. It will take half an hour to stabilize and reach to this 37 degree. So before you start your live cell experiments, at least half an hour before, you should optimize with this, make it on, and then stabilize the entire environment. So once you need, you have to just click a tick, and then this will be active. So humidity, the temperature of the insert, the plate on which the sample is being loaded, and then the CO2. So CO2, if it is at 5%, you can see right now it is at 0.3%. From the cylinder it has to be open, and here you have to monitor it. So once this is open, you can see the changes here. So it is trying to set according to the changes, and it will optimize based on the set parameters. So after half an hour, when everything is ready, we will take the sample for imaging purpose, right? And what we'll have to do is, we can do time series experiment. So time series experiment will do it if you want to do it for repeated number of samples. So we'll just take the same sequence track, we'll optimize first. Okay. Optimize. So this is the signal we are getting. What we'll do, we can change the color to red. So once we have done for track 1, now if you want to do time series, you have to just click on the time. So in the time, here is the window which will come. You can have how many cycles. So I want to repeat how many cycles my experiment want to be. So for example, 10 cycles I want to repeat. And if I want to give an interval in seconds, milliseconds, or whatever time, in hour, days, I can give an interval here. So suppose I give an interval of 10 seconds. So my entire sequence is like 10 cycles. Whatever the time taken for this sequence plus this 10 seconds will be my one sequence time, and it will be repeated for 10 times. So once I have done this, I can start the experiment. But I also have options, like I can have, before starting I can have number of slices acquired. I can do it afterwards also. So pre-acquisition, post-acquisition, how many frames, you can all decide from here. At what time you want to stop, you can also give that, or what to stop. And before you start, you need a manual command to start, that also you can set from here. So options are here. And then you say start experiment. In the start experiment, this is against the time, nothing is going to change. So you can see both the things I have got over a period of time. So in this I can have time series for live cell experiment where I can have for different time frames, and then based on it I can do it. Now there is one more thing which I can also do. In time I can also have, there is one more option called positions. So like experiment regions. So I can go to different experiment regions and then I can select. So for example, I have this is the center, okay. So I am focusing at this region here, and what I'll do is I can simply go and add one like tile I have selected. So in the tile option, you see I can select position. So this position I have selected here, and I can have a 3 by 3 matrix for tile and stitching. So once I have done this, I'll do stitch and it will take a stitched image. So once this frame is done, for red and green, it will go to the next frame and try to stitch with it. Okay, so you see this is for red and green, and if I want to have a stitched image, I'll say start experiment, and similarly I will try to see if I can get a larger field of view with the same information. So you see here, this is red, this is green, and then it will be stitching. So as you can see, this is one frame, this is another frame, 3 by 3 matrix. So 3 by 3, 9 frames per channel it will take to one field of view. So you can have n number of those positions here, and then based on that you can select. Now if you don't want to stitch, you want to have multiple wells and you want to do that, what you have to do is, in that case you have to simply delete this sequence, and in below there is a position. So in position, what you have to do is you select one position, okay, and then suppose you want to go to a different position which is not adjacent to it, right? So for that case, what you do is you have this area, I go to the other position where I don't want to have. So it's altogether a different plane or different field of view, rather, not plane, but field of view. And then I've selected this region, suppose, and I say plus. So these two are separate. You see X and Y, Z is same. So once I do this, I say position. Right. Sorry. So this was 9 tiles. You see here, if I give region, so it was 9 tiles. So here in the first one, it was like 3 by 3, 4 by 4, whatever you want. But if you want to give more, you can also have the options like this. Select one position, point A, select other position, it will stitch everything which comes in between, right? But there is another one, so where, I'll delete this, where you want to mark position, one position, and go to altogether different position. Now, so register these 2 points. So this position I have registered, then I'll move to a different position. So I move to a different position, and then I'll also mark this position. So these are 2 different positions which I have marked, and I want to take images from positions, 2 different positions. So I'll just click, and then position A and position B will come. So this is position 1, let us say 2. So here position 1 and position 2, these are different. Okay, I can have multi-channels, I can have Z-stack, I can have time, everything I can have for different positions also. So if you are using a multi-well plate for well A, well B, well C, whatever it is, You can do it with a different time gap also. You can do for Z, you can do for like different multi fluorophores also. So all this is possible here. And finally there is one more thing which I would like to show is a experiment designer. So you can have design your experiment. So right now this is one experiment or sequence which I have designed only with red channel. Okay. Now if I want to create a second one, so I have created a second one where I want to do a confocal image with a dye. Suppose I want to have something called in green. So I will simply select this and I will take the green dye. I have taken the green dye and I am selecting this window. The laser I am using 488. Okay. And then with this case, if I want to do it, so you see here, once I go to the experiment designer part, you see I have a green here. So if I want to have green as well as I want to have different tracks, so I can have one more track which would be like, if I want to do it simultaneously, I can use a DAPI for instance, and this DAPI also I can select whatever range I want, and I can use this. So you see here, based on this I can have multiple number of sequences and I can design it. In between, if I want to give loop, I can say from 1 and 2 I can want to give loop. I can add loop from here. So you can see the loop, how many loops I want to give. I can give number, start and end. So start with 1, end with 2, and this can be active. So once this is ready, I can have start the experiment, and then based on the sequences which I selected, it will automatically